Hi there, and welcome to the presentation on the generation of whole genome allele-specific maps for CRISPR-Cas9 genome editing. My name is Jacob Bradford, and I present this work with Bo Zhao, Alexander Urban, and Dimitri Perrin. You might be asking, what is CRISPR-Cas9? It is a genome editing technology that we can direct to modify any DNA. To direct Cas9, we need to provide a synthetic guide sequence that is 20 base pairs in length. There are two critical properties that we must consider when designing the guide RNA sequence. The first being efficiency, which looks at how likely is modification to occur, which we also call on-target. The other is specificity, which looks at the risk of modifying elsewhere apart from the desired on-target site. We call this off-target. In a previous study titled Improving CRISPR Guide Design with Consensus Approaches, we explored how to improve the prediction of guide efficiency by combining multiple approaches. Let's take the example where we have 4 existing approaches. We ask the question, how precise is the consensus vote when all 4 approaches agree that the guide would be efficient? Or, how precise is the consensus vote when at least 3 out of the 4 approaches agree that the guide would be efficient? Or, how precise is the consensus vote when at least 2 of the 4 approaches agree that the guide would be efficient? We found that the combination of multiple approaches is more precise than any individual pipeline alone. When we combine the prediction by MM10dB, sgRNA scorer 2, and ChopChop, we get the best precision whilst having an appropriate recall for your experimental needs. We chose to focus on these 3 approaches because in our previous benchmarking study, they were able to scale to large data. But we took this performance-focused consensus vote and re-implemented them into our own pipeline named Crackling. Using this approach and our really fast off-target scoring, which I'll talk about in a moment, we found that Crackling could design at least one guide for 95% of rice genes and at least 3 guides for about 92% of rice genes. For faster off-target scoring, we use a technology named Issl. For this, a candidate guide and off-target sites are sliced 4 times, such that there are 5 slices. To find off-targets that are similar to the candidate guide, we identify slices that the sequences have in common. Imagine that each of these dots represents a targetable site. If the candidate guide and a targetable site differ in 4 or less positions, then we'll consider that targetable site as an off-target. So let's look at slice 1, GGAG, and find all of those potential off-target sites. These are the ones that we find, and a subset of those are real off-targets. By using Issl, we don't need to consider every single targetable site on the genome because they're not all true off-targets, but rather just a subset of those. If we were to look at a more concrete example, it would look something like this. In panel A, we have 5 target sites labeled number 1 through number 5, and for each, we have a count of how many times they reoccur on the genome. Notice that each sequence has been sliced 4 times, such that we have 5 slices. In panel B, you can see that some slices are shared amongst multiple sites. So when we look at slice 5, when it has an identity of CATG, sites 1, 2, 3, and 5 share that in common. If we look at slice number 4, we can discover site number 1 when slice 4 has the identity TATT. We can also identify sites 2, 3, and 5 when slice 4 has an identity of CATT. Here we have a candidate guide C that needs to be assessed for its off-target risk, and we have the same 5 target sites from the previous slide. If we look at slice number 5 for C, which has the identity of CATG, you will notice that there are some arrows on top of slice number 5 for target sites 1, 2, 3, and 5. That means C and those target sites have slice 5 in common. Based upon this, we will assess how many mismatches there are between C and each of those target sites, and if there is 4 or less, then we will score them due to their off-target risk. This plot shows the speed of ISSL compared to others. The horizontal axis is a list of genomes that increase in size. The vertical axis is the runtime for each method to assess the off-target risk of 10,000 candidate guides. As you can see on the smallest genome, which is about 400 megabases, ISSL took 9 seconds, whereas everybody else took about 100 seconds. This trend continues throughout the plot. Now that we know that crackling is very precise due to the consensus vote and very fast due to ISSL, we can start designing whole genome allele-specific maps for CRISPR-Cas9 genome editing. We do this by taking the reference genome and a file that describes allele-specific differences. Using this data, we identify and extract candidate guides. Then, using the consensus vote and off-target scoring, we assess those candidate guides and select only those that we believe to be safe and efficient. The common marmoset is an ideal non-human primate that can be used for engineering objectives. We designed guides for the embryonic stem cell line CJ367. Using the reference and alternative sequences of the marmoset, crackling identified nearly 6,000 safe and efficient gene targeting guides. Using these guides, we successfully targeted CJ367 cells and sustained pluripotency after dead Cas9 transfection. We also induced functional neurons in a rapid and single-step process. I hope that you've enjoyed our presentation. My name is Jacob Bradford. If you'd like to make contact with us, please do so via Dimitri Perrin at dimitri.perrin@qut.edu.au.